For this investigation, we want to look at the enzyme lactase. Notice the ASE ending indicates that it's an enzyme. Enzymes typically end with ASE, in this case lactase. But also remember that enzymes are specific. So lactase will break down lactose. You might remember that lactose is a disaccharide, a double sugar. That means it's made of two parts, a glucose unit and a galactose unit. So if we can get that lactase to break that bond and separate that disaccharide, then we should have some glucose available. Now, why is that important? Well, because we have glucose test strips. So we can test for the presence of glucose. And if we can test for the presence of glucose, we can see if our lactase is actually doing its job. Now, there are commercial versions of lactase, things like Lactaid, and so that's what we're going to test today. Does Lactaid digest lactose? So I have some milk and I have some Lactaid, and we'll work on this experiment. Now, you might be thinking to yourself, well, if a person takes a Lactaid tablet and it goes into their body, it's going to wind up in their stomach. Now, what do you know about the pH of the stomach? Well, you know the pH of the stomach is fairly low. It's a strong acid. So we also want to test, how does that Lactaid perform in the presence of a strong acid? So we're also going to add an acid into our experiment. So the first thing we want to do is have a control. So what we're going to do is fill this well plate with some solutions. We'll put water as our control, and then we'll add across this row a normal set of solutions, milk and glucose and milk plus Lactaid. And then we'll also add some of those things in this row, but we'll add some acid to it. So take a look at how we should set up our well plate. So I have set up my wells. This is my water, and that is for a control. And then I have a well of glucose. Here I have milk. Here I have Lactaid, and here I have milk plus Lactaid. Down here I've added acid to each one. So here it's Lactaid and acid, and then here it's milk and acid, glucose and acid, and then milk, Lactaid, and acid. Now I need to stir the milk and the Lactaid. But if I stir one of these wells, I need to stir all of them, because we need to be certain that it's not because of the stirring that we get a result. Now what I'm doing is using one end of a toothpick to stir, and then I'll flip it over so I don't contaminate my solutions, and I'll stir in one of the other wells. So I'm stirring, then I'm rotating my toothpick, and then I'm stirring, and there I'm stirring, and then rotating my toothpick and stirring. Now I have these glucose test strips. I'm going to add those test strips to each of the wells. So our control first, which is the water, and then I'll add it to the glucose, and if I can grab one, to the milk, and then to the Lactaid, and then to the milk plus Lactaid. Now you might be wondering, how am I going to know when the test strip is working? All right, it's a great question. So these glucose test strips turn green if there's sugar present. So we're looking for a sugar color, a green color. Now we have to wait a little bit for the action of the test strip. So we'll pause for now, and we'll get the results in 5 minutes.